Okay, good morning, everyone. So today's lecture is going to be a little different. So far, we've learned about all of the innate cell subsets, what they do, what their job is, how the T cells and B cells develop. And then this section of the course, we've spent a lot of time looking at how responses get started and what happens to them. And so this lecture is taking all of that and putting it together in sort of a hypothetical immune response. What happens during the response and what's the timing of it? Okay, so the first thing is when you think about a response or an infection, there's several steps that have to happen. And for those of you who haven't taken microbiology or virology, this is sort of a review of what happens during a infection. In this case, I work on viruses, so I'll use a virus as an example, but the same thing happens with bacterial infections. The first thing that happens, we've talked about immune barriers, and so the pathogen has to get past that first layer, either the mucosal epithelium and all of its extra defenses, or the skin epithelium, which really is, very non-supportive of most viral replication. Now, when it does that, it's going to get in and start replicating. And so during that process, it's going to stimulate the PAMP receptors, and the dendritic cells will take up antigen and start presenting it to T cells. The dendritic cells then get those PAMP receptor signals, which tell them, yes, there's something here. And so they migrate to the lymph node where they can stimulate CD8 and CD4 T cells, okay? Now for this to happen then, CD8 T cells proliferate and leave the lymph node and go do their job, where CD4 T cells will also generate follicular helper cells that migrate to the B cell zones. So the T cells, when they leave, will go out and try and kill infected cells, and they'll do this by perforin and granzyme, or the TNF receptor pathways, TNF and Fas ligand, and that allows them to go out and kill target cells. But you also have the B cell response happening, okay? And so for the B cell response, the antigen is transferred to follicular dendritic cells that then present this to B cells, and those get activated in the B cell zones, and they meet up then with the CD4 cells that are the pre-follicular helper CD4 T cells that provide that key CD40 ligand signal, or CD40 to CD40 on the B cell, that gives that B cell full activation. And those B cells then undergo somatic hypermutation, affinity maturation, isotype switching to an appropriate antibody isotype, and then they differentiate into long-lived plasma cells and memory B cells. So we're going to talk about the timing of these different things and how they actually coordinate during an immune response, because this is sort of oversimplified. We've left out all the other innate cells. We've left out the primary foci B cells and the different steps of CD4 activation. So we'll go through those in just a second. And it's useful, I think, to sort of put this in context of profile. And so you have basically an innate response to a pathogen, a T cell response, and a B cell response. But those are actually made up of many different parts of a response. And so we're going to take each of those separately. What I want you to take home from this, though, is that even as the pathogen enters the body, as soon as it gets in, the innate system can see it. And even after you can't detect the pathogen anymore, you see there's still an innate response that's making a response to the leftover bits of this. And so if it's a bacterial infection, even though there's no live bacteria, there's still PAMPs that are derived from that. Okay, so the purple here is the innate response detecting the pathogen. As long as the pathogen is there, the innate response is detecting it. Now, what you might notice is that the T cell response and the B cell responses are delayed compared to the pathogen. Often during many acute infections, you'll have the peak of the T cell response coincides with when the pathogen is being eliminated. And this is why often people will say, well, I'm sick, but I'm not infectious. Well, that's because you're sick mostly from your immune response, but you no longer have the pathogen there. So that's where that term comes from. So we're going to go through each of the different parts of these responses and sort of divide up where we have the different the different parts of the response. Okay, so we know that the innate immune system has two main jobs. One is to provide the initial response to the infection, and that's very important because without that, the pathogen will grow unchecked. The second job is to inform the adaptive response. Hey, there's something here you need to make a response to. So we know this from experiments where you can infect a mouse with a pathogen, and if the mouse lacks, if the mouse is intact and has a proper innate system, you can see that the innate system can oftentimes control an infection on its own. And so you have pathogen growth gets curtailed with innate recognition. But if you have mice that have defects in either recognition or elimination of pathogens, then when you infect them with either bacteria or a virus, then that pathogen is not controlled. And you can see that the pathogen keeps going up. And usually those mice are susceptible to disease and will succumb to it. Okay, so we know the innate system has a very important job for initial defense, as well as informing the adaptive system. So when we're talking about an infection, I think it's useful to review what happens during an infection. And in particular, viral infection is one example, but the same steps typically are happening for bacterial infection. And this is a flu virus, an orthomyxovirus that is getting into a cell and then leaving that cell. And this is often what virologists focus on, but we'll sort of go through the systemic part of this in a bit. But the first step is the virus has to attach to a cell. And usually this is by surface glycoproteins. For influenza, it has the hemagglutinin and neuraminidase proteins that are important for attaching and fusing. And so the virus has to get from outside the cell into the cytoplasm. And so it's taken up in endocytic vesicles, and in the low pH of those endocytic vesicles, that it causes fusion of the viral membrane with the host endosomal membrane and dumps the genetic material into the cytoplasm. Now, flu is a little different. It replicates within the nucleus. Many viruses just replicate in the cytoplasm, but in any case, the first step for a virus is to replicate its genetic material. And that does that so if one, it has more genetic material to make new viruses, and you can have genetic material so you can get transcription and translation. So you have replication of genetic material that then gets expression of viral proteins. Those are then going to assemble at the surface and will cause budding of the virus so it can get back out. Now, there are many steps here where you could have possible innate recognition, right, after release of the virus. The first is during that entry step, right? You have endosomal toll-like receptors, and they're going to primarily recognize nucleic acids. Well, where are those coming from? Well, virus fusion with the endosome is actually a pretty messy process, and so a lot of virus doesn't make it. And so you're dumping genetic material inside the endosome, and that can be recognized by TLR3, 7, 8, and 9. So that's the first step. You could possibly have recognition. But for viruses, mostly what you're recognizing is abnormal levels of viral genomes or expression of viral RNAs that often are capped differently than your RNAs. And so those you're able to sense those with your intracellular PAMP receptors, in particular the Rig-like receptors and the NOD-like receptors. Okay, so there's multiple steps that you can recognize even during that first infection of the cell. So we go back and look at these about where are these different recognition receptors expressed. And so you can recognize microbial PAMPs either on the surface as cells are trying to get into another cell to infect it, or in the endosomal pathways. And so here I've highlighted for viruses in particular, as the virus comes into that endosome, you have all of these different toll-like receptors that can signal the cell, yes, we need to make pro-inflammatory cytokines. And so for many of these toll-like receptors, primarily you're going to signal through the ERF proteins, interferon regulatory factor proteins, giving you type 1 interferons. In an infection, if you're going to make an adaptive immune response, you need one of two cytokines, either type 1 interferons or IL-12. And you get both of those from these toll-like receptor pathways. You also get pro-inflammatory cytokines like TNF and interferon gamma. They're going to be activating other innate cells, but also remember, interferon gamma is going to be shaping the CD4 response. So if you have a viral infection, you're going to have a very strong response, and typically a Th1 response where you need an anti-intracellular immune response or adaptive response. Now, the other side is when you have stimulation of the intracellular cytoplasmic pathways. Okay, here it says cytosolic, but you can cytosolic or cytoplasmic is fine. And there really are two, right? There is, we divide them here. Sorry, that's not quite right. We'll divide them here. So we have the NOD-like receptors on the left, okay? These are going to form the inflammasome. And from the inflammasome, you get activation of IL-1 beta and IL-18. These are potent inflammatory cytokines that really are telling all the other innate cells, hey, there's something here, we need to make an immune response to it. Do you need to know the names of all the NOD-like receptors? No, we're just going to treat them primarily as one kind of, as a monolithic receptor. But there are several. And they can also sense extracellular signals, but the main job is to really provide that inflammatory signal. Now on the right, you have the Rig-like receptors, right? So the prototype is Rig1 or Rig-I, depending how far back you go in the literature. It was originally named Rig1, but now most people call it Rig-I. And from this, you're going to again signal through the ERF proteins to get type 1 interferons. And you're going to signal through other pathways to get IL-12, TNF, and interferon gamma. So from both of these pathways, you're getting very strong Th1 cytokines that are instructing the response. There's something inside the cell, so we need a cytoplasmic response or cytosolic response, which includes lots of CD8 T cells, killer cells, and then appropriate IgG antibodies. So if you think about this, when we talk about the innate response, there's really two different things that are happening here. You want to have recognition by innate immune cells that there's something there. And once they do that, then they'll start making pro-inflammatory cytokines that activate innate cells. And so you get killing of the pathogen or of infected cells. Okay, so the first step is recognition. And then in doing so, you get production of cytokines, which leads to innate cell activation and killing or effector mechanisms. Okay, so they overlap, but there really is a two-step process here. Okay, so we talked about a virus getting into one cell, but I think we all know that viruses don't just stay in one type of cell, they spread throughout the body. And so here again is an influenza virus. It's got hemagglutinin and neuraminidase on the surface, and it's going to attach to the respiratory epithelial cells. Okay, and so once it does that, it can be sensed by innate cells like NK cells and gamma delta T cells, which we haven't spent a lot of time on, but they can recognize infected cells directly. Okay, but once it does that, it's also going to get, once it's going to travel to other sites of the body, it becomes exposed to other parts of the immune system. Okay, so if it just stayed in the respiratory epithelium, it could live there, and many viruses just stay at the site of entry and then they come back out that way, but others will spread to other parts of the body. Okay, so as the virus comes from the respiratory epithelium, it's going to be recognized by macrophages and dendritic cells, and in particular, the dendritic cells that are going to take up virus and cross-present it to T cells are very important for starting that, that adaptive immune response. Okay, so there's macrophages and DCs that are going to take up the virus, and those have the toll-like receptors, and and Rig-like receptors that can sense that there's a virus there. There are also other cells like the ones that we talked about, the plasmacytoid dendritic cells that are going to make, they're the main source of type 1 interferons. Okay, so you have the first site of infection, then as it tries to move throughout the body, you have the second spread of the infection and production of other immune responses. Now, eventually, those dendritic cells will get to the lymph node and they'll start presenting antigen to the adaptive immune system. So that includes macrophages transporting stuff there to hand off to follicular dendritic cells to stimulate B cells. That also includes migratory dendritic cells that are going to pick up antigen in the periphery and go to a lymph node and stimulate T cells, or lymph node resident dendritic cells that pick up antigen that comes in through the follicular reticular network. Okay, and we've covered all of those already. So the important part of this, though, is looking at these innate cytokines that are being made. Here, IL-1 beta. That's a potent inflammatory signal that helps activate B cells. There's things like IL-12 and the interferons. And you sort of see that they're very important for shaping the response as you go along. In particular, interferon gammas are going to give you lots of IgG to control a systemic infection. Okay, so these early steps in shaping the response are important for the outcome of infection, but this is also showing that there's multiple steps giving you a full adaptive immune response. And that's what we're going to focus on, is the timing of how each of those things happen. Okay, so when you break down the innate response, there really are there are some other differences here. And I think this one is just kind of breaking down based on where is the response. And so the first step is that during infection of that respiratory epithelia, so here, sort of talking about what happens there. And so you have infection, and as that virus is replicating, in particular in the cytoplasm, you have production of nucleic acids that the virtually all cells can see. This will alert the NK and gamma delta T cells that, hey, this cell is abnormal, you should try and kill it. Now, in reality, stimulation of the toll-like receptors actually takes a bit longer. And that's because many infections start with a very low level of pathogen. So for example, most sexually transmitted HIV infections start with one virus particle. Okay, so one virus gets in, and that then has to replicate. So one virus is not really enough to stimulate toll-like receptors. You need multiple of those. So the actual first set of stimulation is the intracellular cytoplasmic sensors that are sensing the replication of the nucleic acids. The toll-like receptors don't really kick in until after you've made enough pathogen to sense, okay? So that's the second step, right? The first step is the infection or the virus gets into a cell, and then you get during replication, you get stimulation of the nod-like receptors and Rig-like receptors. That activates the cell to change its expression profile so it can be more easily killed by NK cells and gamma delta T cells. Now, after that first wave of replication, now you do have enough pathogen to stimulate the toll-like receptors. And that's really the important part, or the part that is important for activation of macrophages and dendritic cells. Okay, so there's sort of like a two-step infection scenario. So you have on the one hand, we talked about there was recognition and then killing by effector cells. But if you break it down, the recognition is actually two different phases. Early recognition of nucleic acids that are replicating, followed by TLR recognition of things of the pathogens that have reproduced. So when you look at the types of cytokines that are being made, virtually any cell in the body can make type 1 interferons. Okay, so this is from the Rig-like receptors, you're going to have signaling through ERV3 and ERV7 to give you type 1 interferons. That's important for shutting down all the neighboring cells, right? Poisons all the neighboring cells to get ready. If you get infected, you're going to shut everything down. Now, as the pathogen replicates, then you have recognition by toll-like receptors. And now you get more IL-12 to further activate the immune system. But these are more specialized to innate immune cells. Okay, so not every cell in the body can make IL-12 or TNF or interferon gamma. These are made by innate immune cells. So you really have to have activation of those cells first, and then they can start producing the inflammatory cytokines. Now, when we draw this, if you look back at our reference, this is this purple line going up and down. It's a composite of these two things, right? They're overlapping. And so earlier you get mostly type 1 interferons in the first hours after infection, whereas later you get production of IL-12 and TNF that are going to shape the adaptive response. Okay, so what are these doing? Well, viruses and bacteria will sort of activate different types of cells, okay? And so if we're going to shape the immune response, the first thing that's going to happen is that those early signals are going to shape what kind of dendritic cells you're going to get. Okay, so in particular, if you have a very strong viral or bacterial infection, you're going to get stimulation of lots of PAMP receptors, and that then leads to production of IL-12. Now, one of the main sources of other cytokines are the NK and NKT cells. If you have dendritic cells making IL-12, NK and NKT cells will make interferon gamma. That's going to shape the CD4 T cell response to be a Th1-like response. Okay, so IL-12 and interferon gamma are important for giving a Th1 response. IL-12 sort of starts it, and then interferon gamma is going to sustain it. If instead you have worm, helminth, or parasitic infections, if you think about the number of actual pathogens, it can be one, right? It's a very low burden of infection compared to viruses, which are millions per milliliter of blood. If you have a helminth infection, it's like 1 per person. So it can be quite low. And so instead of getting all these inflammatory cytokines like IL-12 and interferon gamma, you'll get signaling that leads to production of IL-4 by the NKT cells. Okay, so lots of cells can make these cytokines. As we said, they're really part of that second wave of innate cell production of cytokines. That then is the key signal then to shape the CD4 response. If you have IL-4, you're going to primarily make a Th2 type of response. And that's going to give you things like IgE so that you can have your mast cells activated and start killing this, as well as your other granulocytes. Okay, so let's talk about how this starts. How long does it actually take for things to get to a lymph node? And really, when you're talking about how they're getting there, you're really talking about size difference, okay? So when you're talking a typical lymphocyte is about 10 microns or 10,000 nanometers. Macrophages are a little bigger, they're 20 to 30,000 nanometers. Well, those are cell level, okay? Anything smaller than a bacteria, so anything like 1 micrometer or smaller, can freely diffuse to the lymph node. I'll draw the line over here, right? But anything smaller than a bacteria can get to a lymph node on its own. And that's where it's going to go through the follicular reticular network and be picked up by dendritic cells in the lymph node. On the other hand, if it's bigger, for example, bacteria or fungi, large viruses, those are going to be mainly transported by macrophages. So they get coated with complement proteins, and in particular IC3b, that then is recognized by the macrophages, and they transport it to the lymph node. And so that's an active process where the macrophage is entering the follicular reticular network and moving to the lymph node to follow signals to hand this off to the follicular dendritic cells. Okay, so they do overlap quite a bit. So in this range, you can see you can have either free diffusion or cell transport. But as the bigger it is, the more important that the macrophage transport is. So when you look at how fast can antigen get there, this is just small antigens, so protein antigens. And they're looking at how much is actually getting into from the foot pad of a mouse into the draining lymph node, which is a popliteal lymph node behind the knee. And so they inject ovalbumin, the deadly egg protein, right, that has been used as model antigen for a long time. And you can see that it's getting to the lymph node within 3 hours. Okay, and that's with or without adjuvant. Adjuvants are just things that trigger toll-like receptors or NOD-like receptors or RIG-like receptors. In this case, it's LPS, so it's triggering TLR4. And you can see that the timing is the same, but you get a lot more there. And this is likely due to cell transport, increases in lymphatic permeability. So it's actively opening up the lymphatics to get the adjuvant there. Well, within a couple hours, you can see it in the lymph node. You can also do this with fluorescent dyes, where you inject a fluorescent dye, which is a small molecule, so it's going to diffuse very easily. And within hours, you can actually see that fluorescent dye coming in through the afferent lymphatics into the lymph node. It accumulates sort of around the trabecula in that subcapsular space. Okay, so it's only going to enter into it or surrounding the B cell zones in that subcapsular space because it's not a protein that can be transported by those subcapsular space macrophages. Okay, so antigen can get there within hours, and even if it's cell transported, it can still get there very fast. So as soon as you get an infection, it's going to be transported to the lymph node to try and generate an adaptive response. The cellular transport is a little slower, right? This is where a macrophage is taking up something that's been killed by complement in the periphery and then transporting it to the lymph node. And this is a case where the inflammation signals are much more important, okay? So when you think about this, it's a little slower for two reasons. One, you have to have activation of the complement cascade and you got to kill the thing, right? And then inactivation of C3b to IC3b. So that can bind to a macrophage and it can transport it. On the other hand, so that's the first step. On the other hand, you're talking about cytokine production that then activates those macrophages and dendritic cells and tells them to go to a lymph node. Okay, so there's two steps. You got to kill it and have something to transport, or you got to get the inflammatory signals. So that takes some time. So when you're doing this, you can see that this is just an example of dendritic cells. The dendritic cells have to come in, their virus is coming in, it's getting into the cells, and cells are pinocytosing it. Then you have production of inflammatory cytokines that leads to them going to the lymphatics. This happens a lot more if you have inflammatory signals. So we often give adjuvants. In this case, you're painting the skin with this fluorescent dye, and you're providing a toll-like receptor signal, and that increases the speed of this. And so within hours, you have dendritic cells being activated and moving to the lymphatics to get into the lymph node. Okay, so for free transport, you're talking a couple of hours, 3 hours for antigen to get there. That's for free diffusion. For cellular transport, you have these additional steps of cytokine production and informing them, yeah, you should go to a lymph node. So it takes a bit longer. And so you can see about 3 hours versus 18 hours. Okay, so that's really what I want you to know about the innate response, is that you're getting these different levels. You're getting, as the pathogen is replicating, it's generating stronger and stronger signals that are being recognized by these innate PAMP receptors. But now the antigen's in the lymph node, and you're going to have to make a response to it. And so we'll start off with the B cell response, right? And we've sort of put this all together here. Here's our subcapsular space macrophages. They're actually going to pass off antigen to the FDCs. The B cells are going to be activated on the FDCs and head towards the T cell zone, okay? So there's initial B cell activation that we've talked about, and then they go looking for help. If they do not get the T cell help, okay, so In this case, they don't see pre-follicular helper cells. They'll keep going into the T cell zone, form the non-germinal cell plasma cells, plasmablasts, and the primary foci. Okay, so this is the primary foci. That's your initial wave of antibodies. Well, what kind of antibodies? Well, there's no CD40 signal, so it's primarily going to be IgM, and that's very good at further activating complement to really give you more antigen coming in here. If it does get T cell help here, right? Then those B cells will form a germinal center. They will initially go to the dark zone where they proliferate and start doing somatic hypermutation and isotype switching. They'll then go to the light zone where they're then going to be selected for the best cells. And then you have, once the antigen is gone, now you're going to have the B cells that are fully differentiated to become long-lived plasma cells in the bone marrow, or memory B cells that are trafficking just like a naive cell. Okay, so as this is happening, that's a B cell response, but remember you have a concurrent T cell response. Here's our antigen prime dendritic cell, so it's gotten signals to go to the lymph node. It activates the helper cells and as well as the CD8 cells. As those proliferate, this is going to come up and be able to stimulate B cells. But from that, you also get follicular helper cells. Remember, if it's a Th1 biased response, you get Th1 biased follicular helper cells. But then you get the effector cells. So here's our Th1 cells. Those leave the lymph node and they go and do their job. Okay, so we're first going to just focus on these three different waves of the B cell response. Our primary foci plasmablasts, our long-lived plasma cells, and our memory B cells. And so when you look at just the antibody response, it really comes in two waves. You have the first wave, which is earlier, and that's from the primary foci. That's going to be mostly IgM to facilitate further complement activation. And then as you get long-lived plasma cells that move to the bone marrow to just make antibodies for the rest of life, there is a second level of antibodies. And depending on the infection, you may have heard, looks like coronavirus antibody responses are waning. In reality, this often goes up and sort of comes back down to a sustainable level. We don't do that in this class just because it makes it, there's too many different death phases here. In general, you just make a second wave that then that remains stable for the rest of your life. Okay, so when we go back to the B cell activation, it's important to remember where those two waves come from. So here's initially our B cell getting activated by the B cell receptor and the co-receptor, complement receptor 2, recognizing antigen IC3B on the follicular dendritic cell. Okay, that will, first of all, give you short-lived primary foci plasmablasts. That's the first wave of the antibody. But remember, the other thing that when they're trying to do this is they're trying to go towards the T cells to look for T cell help. So here's our T cell being activated on a mature dendritic cell. Okay, pink cell here. That then tells it to go towards the B cells zones and hopefully provide the CD40 ligand signal to the B cell. If it does that, that's where you get the second wave of antibody, as well as memory B cells. But you get the second wave of antibody comes from those long-lived plasma cells that are germinal center dependent. So when we break this up, there really are two waves to the antibody responses. I'm showing them in green and yellow here. So the blue line is a composite of those two, but you can see the green here is that first burst of mostly IgM antibodies from the primary foci. And that's short-lived, it goes away after a few weeks. The yellow is the sustained long-term antibody production. That's the long-lived plasma cells that are going to set up residency in the bone marrow and pump out antibodies for the rest of their life. But there's a third wave here that we didn't talk too much about, and that's the memory B cells. Okay, so memory B cells are going to be exiting the germinal center about the same time as the long-lived plasma cells, and their job is just to circulate and look for antigen for the next response. If you get infected with the same thing, you have preformed memory B cells that then are going to be able to respond faster and higher magnitude the second time. Now, having said that, one of the things we need to keep in mind is that the follicular helper cells are influencing the B cells, okay? And there's lots of different ways that they're doing this. Primarily, it's by cytokines, right? Th1 cells will make interferon gamma to sustain a Th1 response. Th2 cells will make IL-4. Then you have the other ones, the Th17 cells that are making IL-17. And then you can have sort of regulatory cells that are shaping this. The main ones that I want you to focus on are the Th1, Th2, and Th17. The other ones are sort of, they're in between. An IL-21 producing Th cell is sort of in between a Th1 and Th17. An IL-10 producing T helper cell is sort of in between a Th2 and a regulatory cell. And those are fine distinctions that I don't think are really that important for this course. So they're making cytokines. They're also providing these other signals, right? We talked about the other co-stimulatory signals, OX40, ICOS, and others that are telling that B cell what to do. And primarily some of these are saying, become a memory B cell or become a plasma B cell, okay? So, and that really is turning on different transcriptional programs that differentiate these cells. The main ones for memory B cells is a transcription factor called BCL6. That's a master transcription factor that controls those cells becoming a memory B cell, whereas plasma cells turn on a transcription factor called BLIMP1. It's essentially shutting everything else down and saying, just make antibodies. That's your main job here. Okay, so the follicular helper cells are influencing the B cells, first of all, which type of isotype to make, but then these other signals are helping them determine, should it become a memory B cell or a plasma B cell? Okay, so when we look at the isotypes that you get, in the primary foci response, you're mainly getting IgM, because what you're trying to do is further enhance the activation of the response, right? So you're trying to get more cells into the response by complement activation. On the other hand, if you have the second wave of the antibody response is after the B cells have undergone isotype switching. And so you get mainly IgG, IgA, or IgE. And that's true for both the long-lived plasma cells, sorry, the long-lived plasma cells and the memory B cells, okay, because they're both isotype switching. And you want, as you're tailoring this response, even for the memory B cells, you want them to be pre-programmed to make the right kind of antibody should you get infected again. Now, this is often seen clinically when you're trying to diagnose people with, was this your first time having this infection, or is this the second time you've had an infection? And one of the main examples of that is for dengue virus, where the first infection is often milder than a second infection due to antibody enhancement of infection rather than protection. And so what people will look at is if you have anti-dengue antibodies, the ratio of IgM to IgG. If it's mostly IgM, that's your first infection. If it's mostly IgG, then that indicates it's a second infection, or it's a later during the first infection. Okay, so that can tell you a little bit about the response. Okay, so that's really dividing up the B cell response. You have initial activation, and then primary foci. Then as you form the germinal center, you get isotype switching and memory B cells and plasma cells, as well as improving the response by somatic hypermutation and affinity maturation. When you look at the T cell response, it's a little different, okay? And part of that is because it definitely has 3 phases to it. So it has an expansion phase, right? That there's rapid proliferation, and then it's generating all of your effector cells. And then there's a death phase where most of those cells are going to die off. And then you have a stable memory phase where the cells are just circulating like the memory T cells are circulating. And this is broken up a bit like the B cell response. And I'll go through that in just a second. So when you have activation of the T cells in the lymph node, right, here's our dendritic cell in the lymph node, it's activating these T cells. These T cells get programmed or told where to go. And so they're getting signals from dendritic cells. So we talked about vitamin A derivatives. The dendritic cell will pass those off to T cells. That tells the T cell you need to go to a mucosal site. So it'll express things like alpha-4 beta-7 integrin, chemokine receptor 9. Those are the tools that it needs to get into the mucosal subepithelium. If it gets other signals, for example, skin signals, right, it will use vitamin D to signal the T cell, oh, you should probably go back to the skin. So it'll express different sets of adhesion molecules and chemokine receptors. Those are the tools it needs to go back into the skin. Now, in reality, your immune system really treats there are two main compartments. It's the mucosal epithelia and the skin epithelia, because that's how everything gets in. It's got to be one of those two. There are some variations where it can sort of tailor this to go to the lungs or the neural system. But in reality, the main two are just either, is it a mucosal infection or is it a skin epithelial infection? Those are the two main pathways. However it works, T cells have to go from the lymph node out into the blood and then get into those tissues. So how fast does this happen? Well, when you look at the actual number of T cells that you can count in a mouse, and so these are T cells that have a luciferase gene in them, so you can actually visualize them over time in an alive mouse. So this is day -1 before infection. Then you infect the mouse, and you're looking for how fast or where are the T cells going and how fast they're getting there. And you can see that the peak of the response is 5 to 7 days out. So in a mouse, peak of the response is about a week after infection. In humans, it's a little bit even longer than that. It's typically 10 to 14 days. So why? Well, it just takes longer for stuff to move around a human. The mouse is much smaller, so diffusion distance is much smaller. So you can sort of see in the draining lymph node, in this case, what they're doing is injecting ovalbumin into foot pad of this mouse. They're looking at the draining lymph node, which is this bright red area that indicates there's lots of cells there. That is saying, okay, the antigen got there. This is just a protein. The antigen got there within 3 hours. But the T cell response antigen has to get presented by dendritic cells. T cells have to recognize that, and then they actually have to go to the site of infection. And so when you're looking at those different factors, the draining lymph node on that side, you can see a response happening, and it peaks about a week after infection, or actually injection of the antigen. You look at the other lymph node on the other side of the body, there's not a response there because the antigen's not going into that other lymph node. Okay, so as they're doing this, they're proliferating in the lymph node, and then they're leaving the lymph node to go to the injection site. And that's really the peak of the response is a little bit delayed compared to the lymph node response. After they do that, they move to the infection site or the injection site where they're trying to clean up. They're killing infected cells or trying to control the infection at that site. So when we talk about a T cell response, we divide it up into these 3 phases. Okay, and this is true for both CD4 T cells and for CD8 T cells. There's the expansion phase. That's going to occur for a mouse in the first 7 days after infection. In a human, it takes about 10 to 14 days. And then there's a contraction phase. okay, where most of them die off. And it's between 90 to 99%. This would be, so if this is a magnitude scale, this would be a logarithmic scale where most of the cells are dying off. But you can see, you end up with a memory population that is higher than your baseline, okay? That's so that next time you get that infection, you can make a faster response that's already selected for antigen-specific cells. So one of the things that was discovered roughly about 10 years ago was that this contraction phase is pre-programmed. And it's pre-programmed to happen whether you control the infection or not. If you do control the infection, great. The immune response shuts off and you have stable memory. If you don't control the infection, the immune system still shuts off, because if it didn't, it would kill you. And so that was really discovered by Vladimir Budovanec and John Hardy at University of Iowa. And they showed that the death of that effector cells is independent of clearance of the infection. And Sukhek and Rafi Ahmed did similar studies when they were looking for memory precursors, but it was really, that is a pre-programmed death, because if you don't shut that T cell response off, the host will die because it's just too potent. The way that they did it, that Vladimir and John did it, was they would use antibiotics. And so if you inject a mouse with Listeria monocytogenes, their sort of favorite pathogen, it replicates inside of cells, and so it acts more like a virus than a bacteria. If you don't treat it with antibiotics, the infection is not cleared and the effector T cells die off. If you do treat it with antibiotics, the infection is cleared and the effector T cells still die off. Okay, so whether or not that infection gets controlled doesn't matter. You shut the T cell response off. And this is one of the ways that your immune system is trying to protect you from its lethal response. Okay, so we know that the contraction phase of the T cell response is pre-programmed to happen. But the question is, when do you generate memory cells? Okay, and so Sukhek she was a postdoc with Rafi Ahmed at Emory, showed that memory cells, or at least precursors to memory cells, are present even during that effector phase of the T cell response. And you can find these, one of their main claims here is that they were actually able to find these by expression of the IL-7 receptor high affinity version. So what they would do is they'd take mice that have T cells specific for a virus, they inject the virus into those mice, and then they collect the IL-7 receptor high cells and the IL-7 receptor low cells by flow cytometry sorting. And then they inject those into other mice. So here, IL-7 receptor high cells come in versus IL-7 receptor low. And then they challenge those mice again with virus. Okay, and they look at which cells expanded, which cells were giving you memory cells. And so what they found is only the IL-7 receptor high cells provided a memory response, okay? Whereas the IL-7 receptor low cells, those were all effector cells and they died off, and so they didn't give you any memory. Okay, so the main point of this is that even during that early part of the response, where the T cells are actively going out and trying to eliminate the infection, you're still getting memory cells even early during that response. And so if you
Dr. Blattman?
Yes.
Back to the clearance of effector T cells. Do we know what gene or cytokine signaling is responsible for this?
It turns out to be they're expressing, depending on which cell type you're talking about, that the master transcription factors are actually turning these cells off. So as they get activated, they start expressing death receptors that then they signal to each other and they start killing each other off. But it's actually not known how a memory cell escapes that, other than by IL-7 receptor expression. But it doesn't appear to be a single gene. There are some mice, the LPR and FAST mice, that the response doesn't shut off. And so it can be partly due to death receptor proteins or death receptor like proteins like programmed death receptor 1, which is where it got its name, as well as the fast proteins.
Thank you.
Okay, so when we break down the T cell response, it too is a composite of sort of two different responses. And this is very analogous to the short-lived plasmablasts for B cells in the primary foci. And then from the germinal center, you get long-lived memory B cells. Well, the same thing's happening for T cells. You get lots of effector cells, right, that are mostly going to die off. But even early during response, you're also getting IL-7 receptor high cells that become your self-renewing memory B cells. Okay, so there's really a T cell response is two waves. You have terminally differentiated effector cells that are all going to die off, and memory cells that provide, or memory precursors that provide your long-lived memory cells. Now, there's actually two different kinds of memory cells, and Dave Massapost and others sort of really flesh this out. But here we have some antigen coming in. You get activation of a dendritic cell, goes to a lymph node, it activates our T cells, and you get terminal effector cells, but you also get memory cells, okay? Now, the terminal effector cells are all going to die off, but you have actually two different kinds of memory cells. So in particular, what's thought is that if a T cell gets CD4 help in the form of IL-2 and CD40 signals, then it becomes a central memory T cell. And the central memory T cells they're acting like the memory B cells. They're migrating from the blood back through the lymph node, okay? We're going back to the blood, looking for an infection. And what the difference is that central memory cells have a high ability for proliferation once they see antigen. They're not immediately effectors, but they can proliferate to generate a second effector response. On the other hand, you have effector memory T cells, okay? And these are going to go into the tissues and sit and just wait for the infection to come in. So once an infection comes back in, they're there in the right place and they're ready to respond. And they have many of the same properties of an effector cell, but they're not recirculating like a central memory cell. They're just sitting out in the tissues, ready to fight off infection. And so they don't proliferate much during a second infection. It's only the central memory cells that proliferate. Now, one of the things that if you look at this, the central memory cells are expressing CD62 ligand, okay? Or sorry, not CD62 ligand, sorry, L-selectin, CD62L. So it's been an early morning. What do we need that for? What does L-selectin bind to? It's a selectin, so it's going to bind sugars, but it binds specifically to which sugar? Anybody can answer.
I'm actually not sure of how to pronounce it, but I thought you said something like Sialyl-Lewis X.
Yeah, it's a Sialyl-Lewis X, okay, which is only expressed in the high endothelial venules. So because the effector memory cells don't have to go through the lymph nodes, they don't express it, okay? So they're gonna have other things that are keeping them out in the periphery, okay? They're also not expressing the chemokine receptor CCR7. Central memory cells, you need CCR7, right? That is going to activate LFA1, allowing cells to get into high endothelial venule. But these cells are not going through high endothelial venules, so they're CCR7 negative. They have other adhesion proteins and chemokine receptors that allow them to get into the peripheral tissue, but they don't need the ones that you go back that you would get into a lymph node with. So different kinds of adhesion molecules. And this is really be effective. They have very different jobs. Effector memory cells are going to provide sort of the first T cell response to a subsequent infection, whereas the central memory cells are going to proliferate in response to a second infection and generate another effector response.
Dr. Blattman,
Yes.
what about CD44? Because I think I've seen a lot of times when they want to do like a staining to show activation of T cells, it'll be like CD62L low, CD44 high. Does that come in here?
Well, all memory cells will have CD44 on them in a mouse. In humans, they typically use CD45 differential transcripts. So if you were to look at CD44, it would be on both of these cell populations, right? It would be on the effector memory cells. It would be on central memory cells. It's also on terminally differentiated effector cells. So any cell that's gotten an antigen stimulus will express CD44.
Thank you.
Okay, so one of the big mysteries is how do you get memory T cells versus effector T cells, right? How does a T cell know which to do? And one of the first ideas was that they're just derived from two different populations, okay? So you have one T cell gets stimulated a certain way, that becomes a memory cell. Another T cell gets stimulated a different way, becomes an effector cell that's going to die off. This has actually been disproven, both by looking at the T cell receptors and showing they're the same, even at a nucleotide sequence level, and by using T cells that all have the same receptor on them. So the effector and memory cells come from the same naive precursor. So this is wrong, okay? It still appears sometimes, but it just is, there's no scientific data that supports this anymore. The second model, which is sort of true, but is not sufficient on its own to differentiate these two, is the decreasing potential model. So this is where if you get a little bit of stimulus, right? A little bit of stimulus, you get a central memory cell, you get a little more, you get an effector memory cell, and then as you get more and more signals, you get terminally differentiated effector cells. The problem with this is that if you actually have mice that skip these signals and go directly to here, you can generate these and just skip all of the ones in between. So it's unlikely that that's the only part of this. We do know that continued stimulation does drive things towards an effector, terminal effector phenotype, but that's only part of the story. So then we come to, well, the signal strength, and this is very similar, that you have a weak signal, you have central memory cells, and if you have a very strong signal, you get terminally differentiated. Sort of in between there, you get effector memory cells. And that also is partly true. You can see that in some settings, but the problem is that those don't account for, you can get a very strong response and still get lots of central memory cells. So it doesn't really fulfill the criteria for that's the way that it gets it. So this one's wrong. We'll give you this one a half a wrong, and this one's a half a wrong. We knew that there are parts of that are important. But what we do know is that this one is probably the most correct. And this was shown by Steve Reiner and others at University of Pennsylvania, where what's happening is there's asymmetric division of the T cells. So as they're proliferating in the lymph node, one daughter cell will get all the transcription factors and the other one doesn't, okay? What that leads to is the one that got the right transcription factors, it becomes a memory cell, and ones that didn't get the right transcription factors become a terminally differentiated effector cell. So we know that that's part of it. What we don't know is how that plays with these other models. Is there some strength of that stimulus or duration of stimulus that is influencing how much of the transcription factors are being passed to one cell versus the other? We don't know it, but this is really the correct model. It was really only shown within the last 10 years or so. but it's not exclusive of these other things having an influence. Okay, so that one's wrong. Those ones are mostly wrong. This one's sort of right, but this one gets all of the right boxes. Okay, so when we talk about the memory T cells or T cell responses, we've talked a lot about different things that are controlling this. So for example, memory cells are IL-7 receptor positive, effector cells are not. Okay, so those are terminally differentiated. Memory cells or central memory cells are going to express CD62L because they need to go back into a lymph node. But a terminally differentiated effector cell, it needs to go out and do its job. You know, if you got infected in your big toe, it needs to go to the big toe. So it's not going to express the L-selectin, which helps it get into a lymph node. Now in the middle, we have these sort of effector memory cells. And some people will divide these into subgroups. We don't do that in this class. But you can look at, they sort of are a little bit of both. They're IL-7 receptor high because they are memory cells, but they're L-selectin low because they're not going through a lymph node. Okay, so they're going to go out in the periphery and hang out there. When you're looking at other transcription factors, and this is true for Th1 type cells. I don't want you to focus on the rest of this. I just want you to focus on the central memory cells express a transcription factor called eomesodermin. Okay, but as they get stimulated or as they differentiate into terminally differentiated cells, they express a transcription factor called TBET. And that really controls all these other genes that I don't need you to worry about. But it's influencing as the cells proliferate and differentiate more, it's telling them what job they should do. Because TBET is going to control expression of these, shut off IL-7 receptor and CD62L, where eomesodermin is going to control these and continue to express IL-7 receptor and L-selectin. So then we came to helpless memory cells. And this was, Joe Sun did it when he was in Mike Bevan's group at the University of Washington. And this really showed what the effect of CD4 T cell help is on other T cells. And in particular, they were looking at CD8 T cells that got primed with and without T cell help or CD4 help. The way they did this is they took wild type mice or mice that don't have MHC class II. If you don't have MHC class II, you don't develop any CD4 T cells. Okay, so it's essentially a CD4 negative mouse. Then they immunized them with listeria, expressing an antigen, in this case, ovalbumin. Okay, so this is going to give you very strong activation of HAMP receptors, so you don't need CD4 help in activating CD8 cells. And so this is really just comparing what happens to the memory cells that didn't get CD4 help. So this had no impact on the terminally differentiated effector cells. So this is ovalbumin CD8 T cells at the peak of the response, 7 days, right? That's the peak of the T cell response after injecting ovalbumin. But then when they re-challenged the mice, long after infection, they found out that, well, you don't have any memory cells. And really what they found is that there were memory cells there, but they didn't proliferate. And so essentially what they're saying is that without CD4 help, we're not getting central memory T cells. So that really that signal for central memory cell is based on CD4 help in the form of IL-2 and CD40 ligand signals. Okay, so we're going to go all the way back to the very first lecture, right? And talk about Thucydides, who in 429 BC, roughly, noticed during the plague of Athens that those who had recovered from the disease were able now to treat others and handle other patients without any fear for themselves. So they were immune. This was really one of the first descriptions of immunity to disease. So what does that mean, right? It's important. And as we talk about in the next section of the course, when we talk about vaccination, we're trying to develop immunity. And so what does that really entail? Well, there's a number of things, okay? The first is you've already got antibodies to whatever the infection was, okay? So that right off the top means that you're gonna have less pathogen. That's giving your immune system a head start. Less pathogen means it's easier to control. Well, because of that, you also have less of an innate response, right? The innate response is proportionate to the amount of infection. So if you don't have a strong innate response, you don't have all the inflammation, you actually have less disease. Okay, so the antibodies are helping control and how much gets in and how much is going to be able to infect you. But then that also results in less innate response activation and therefore less disease. But then we know that we have memory B cells and memory T cells. And what are they doing? Well, you have for the memory B cells, you already have many more B cells that have a high affinity B cell receptor for that pathogen. It's already switched to the proper isotype. So it doesn't take as long for you to get a secondary antibody response. Okay, it's much faster. That also helps with controlling pathogen, therefore reducing the pathogen burden and as well as disease. When we talk about T cells, you have those two populations of memory T cells. You have effector memory cells that are sitting in your big toe, waiting for you to stub it on a tetanus infected knife or something. And so they can provide the initial response at the site of infection that can immediately control it. But you also have central memory cells, and these are selected for higher affinities or selected for the best T cells. They don't require all of the checks and balances to generate a response, and so they have a faster and higher magnitude T cell response. Okay, so you get everything happening faster as well as lower pathogen load. That means that you're really getting a jump start on the infection and controlling it much faster. Many people incorrectly think if you're immune, you don't get infected. It's absolutely not true. If you've gotten a vaccine against something, you can still get infected, but you're just controlling it so fast that you don't get sick from it. You don't get disease. Okay, so what are these antibodies doing? Well, the first thing is you've already got a lot of antibodies against that pathogen, so it's either binding to it and fixing complement, so that's getting everything started faster. It can opsonize it, so macrophages can take it up. For viruses, you have neutralizing antibodies. Essentially, all of those proteins on the virus surface are spring-loaded things that are helping it to fuse to the plasma membrane. So the antibodies can trigger that and neutralize the virus. And for some other things, it can actually cause agglutination, which makes it just stick together, and so it's more easily controlled. Okay, so then we talk about these the recall responses. A naive B cell recognizes an antigen, right? And then it's going to get signals that then it's got to go look for T cell help, come back and form a germinal center. But that takes a while. But if your B cell is going to a memory B cell combined to the antigen, and it automatically becomes can become automatically an antibody producing cell, similar to a primary foci, but it's got high affinity antibodies, and they're also of the other isotypes, right? So they're a better antibody and the right isotype for that infection. Okay, so memory B cells will do that. They also don't require CD4 help to go to form a germinal center. A memory B cell skips that. Doesn't need CD4 help, just automatically goes to the germinal center and starts forming the dark zone and doing their thing there. Memory T cells are also much faster at this, right? And so if you have a tissue resident effector memory cell, it's just basically waiting out in the periphery for the infection to come in. The dendritic cell is going to present the antigen. It becomes activated and makes all of the cytokines it needs to effectively control the infection. Effectively control the infection. Central memory cell takes a little bit longer, right? It has to undergo proliferation, okay? But on the other hand, it again, once it proliferates, can make all of these other cytokines. And so it generates a second effector response. In this case, the main difference is these cells that are sitting out in the periphery, sitting in your big toe, don't proliferate as much. Your central memory cells will proliferate a lot to give you that big burst of effector cells. Now there's some differences here that memory T cells don't require all the checks and balances, okay? When you look at initial activation of a naive T cell, they're really the only cells that can do this to start off a response are dendritic cells, okay? If you have an activated B cell, well, you had to activate it with cytokines and a number of other things. Yes, it can start a response, but where would that have come from in the beginning? You would have to have a T cell response to make those cytokines to mature the B cells to become activated. So that doesn't really work. So only dendritic cells will activate naive cells. But then you look at, well, basically any kind of cell can activate a memory T cell. You can do resting B cells, you can do epithelial cells, almost any kind of cell, as long as it presents antigen, it's all the T cell needs to make a response. Okay, now what is the point of all of this? Well, when you're talking about your first infection with whatever pathogen, first time you've got valley fever, you get all of these different parts of the immune response going, and their job is to control it. Well, what's the point of memory? It's not to prevent infection, it's to prevent disease. And so that's really one of those big public misconceptions, is that it's sterilizing immunity. It's not. It is just to control things faster so you have less disease. Okay, so we'll end a couple of minutes early and I'll take questions, but I just want to remind everybody, the exam is this coming Thursday at 9 a.m. All the same rules and regulations. It's open book, open notes, and you do not require the lockdown browser. So I'll take questions.
Dr. Blattman.
Yes.
So when we were talking about the formation of the primary foci, you said that so when they go to the cortical paracortical junction to meet up with their follicular helper cells, if they get the recognition, if they get the help, then they'll form the germinal center. But if they don't, then they kind of just go past it. Is it possible that one of the follicular helper cells is there to present its antigen, but the B cell just completely misses it and still forms that primary foci? Or are there certain signals that kind of direct it towards that cell or the helper cell?
That's absolutely correct. So you could have one follicular helper cell there and it just missed it. Mm-hmm. Sort of like the Cowboys offensive line. Right. And so yes, absolutely. If it misses it, it keeps going.
Okay, thank you.
Dr. Blattman?
Yes.
When a naive B cell initially gets its antigen and gets the proper signaling to become a centroblast, and then it goes through affinity maturation. When it's going through affinity maturation, is it getting presented the same antigen that it was initially recognized? And if so, how does that happen? Like, how does it know to find a cell with the same antigen?
Remember, it's seeing structural antigens. And so it's just looking for the same thing. So if it was there on the first time, it's likely to be there the second time. So let's say you're a B cell and you recognize a football shape. Well, the antigen is being held on follicular dendritic cells. So after you've mutated your receptor, you're interacting with the same antigen. Whether that's, and if you don't see the football shape, then that likely means that you mutated away from a good receptor to a bad receptor.
Gotcha.
Another way of looking at that is that if a follicular dendritic cell is holding up the carcass of a dead bacteria on its surface, well, the B cell is likely that still got all the epitopes that initially activated the B cell, so it's likely that the B cell can still see it.
And I had one more question. It says that CCR7 is a chemokine, but it's
Chemokine
also
receptor.
a chemokine receptor, but it says also like a cytokine that attracts cells to the high endothelial venule. So is it also secreted?
No. So CCR7 recognizes CCL19 and CCL21. And those are activating CCR7. It's allowing the cells, right? CCR7 signals through a G protein coupled receptor to activate LFA1 to the high affinity form. That's its only job. It's not being secreted or recruiting other cells. It is telling the cell to respond to the recruitment signal.
Dr. Blattman,
Yes.
I also have a question that I probably should have asked a while ago. It's related to one of the previous lectures. So talking about B cell activation, with the cross-linking, when we're talking about antigen bound to IC3B, I thought that IC3B was like the like when the complement proteins cleave C3B, and then I thought IC3B was like an inactive form. Can you
inactive in that it's no longer hydrolyzing. So it still has a role. Its job is still to be recognized by CR2, complement receptor 2, and take whatever it's stuck to and transport it. But it's inactive in the sense it's not going to further activate the complement cascade.
Okay, but it will still bind to antigen.
Yeah, by the time it's IC3B, it's already bound to antigen covalently.
Oh, okay.
So it's C3B gets bound to antigen, covalently makes a bond, and then it gets inactivated to IC3B, but still stuck to it.
Okay, so then that complex then can bind a BCR and the co-receptor.
Correct.
Okay. Okay, thank you.
Dr. Blattman, I have a quick question. So in the previous lecture, we mentioned that Th17 T cells produce like a lot of cytokines, but I kind of got the idea that it mainly produces IL-6. But in this lecture, you just mentioned that it produces IL-17. So which one should I know it as?
Well, IL-6 causes them to become Th17 cells, and then they'll make more IL-6 to sustain it. But they'll also make other cytokines like IL-17 and IL-21 and a number of others. So, but the one that is telling it to be a Th17 cell is IL-6.
Right, okay.
Dr. Blattman, I have a clarifying question also from the B cell lecture. Are FDCs involved in the transfer of antigen to the B cell zone in both mechanisms of antigen entry?
So the FDCs are the terminal site for antigen, right? Or the only thing that can steal it from an FDC is an antigen-specific B cell.
Okay.
So whether the FDCs are getting it, whether a macrophage is transporting it in, and goes through subcapsular space macrophages on these non-antigen specific B cells and ends up on an FDC. The other way is it diffuses through the follicular reticular network, comes out of those leaky pipes, and the FDCs pick it up.
Oh, okay, so FTCs are always there to pick it up.
Yeah. Yeah.
Okay, thank you.
Okay, if there's no other questions, we'll see you on Thursday.